Hello everyone, and welcome to this fourth module of the European Pharmacopoeia training, which will deal with reference standards. My screen is not, yeah. This first presentation will be on how we establish those reference standards and how you can use them. My name is Bert Blanchard, and I work as a study director in the laboratory department of the EDQM. And together with my colleagues, we are responsible for the establishment of the European Pharmacopoeia reference standards. I will start with a short introduction explaining some terms and definitions that we use for the different types of reference standards. Then I will go on to the main part, so the establishment and use. And then I will give a short overview of reference standards that we have for general chapters. And finally, I will briefly touch upon the subject of secondary standards. So the definitions. Before using our reference standards, I recommend that you take a look at the general chapter 5.12 on reference standards. It's only there for information, but it contains all of the definitions that I will show in the next slides. So I might go a bit fast here, but you can always consult them again. It also gives information on how to use, store, and treat our reference standards. So, starting with the general term for a reference material, it should be sufficiently homogeneous and stable with respect to one or more specified properties and is established for the intended purpose. But you can note that this is both quantitative and qualitative. A certified reference material, or a CRM, is a reference material that is provided with a certificate stating the value of the specified property, but also its associated uncertainty, and importantly, a statement on the metrological traceability. A primary measurement standard is established without reference to another standard in the same for the same property or quantity. So it's really established on its own independently, which is of course in contrast to a secondary measurement standard, which is established against a primary for the same property or quantity. The European Pharmacopoeia reference standards are established by the EDQM and adopted by the European Pharmacopoeia Commission, and these can either be and mostly are substances, but could also be preparations or reference spectra. And finally, this presentation will mainly be focused on the CRS, the chemical reference substances, which can either be a substance but also a mixture of substances, which is established for the intended purpose, as stated in the European Pharmacopoeia. And this is really a very important concept that our reference standards are only established for the intended purpose. And then finally, an important concept in the general notices that the European Pharmacopoeia Commission establishes the reference standards, which are alone authoritative in case of arbitration. All right, and then we move on to the establishment and use of the reference standards. And as I just said, so they are intended for they are established for the intended purpose only, which means that the establishment is tailored according to the intended use. To make this clear, I've divided this part of the presentation into 4 categories. So on the one hand, we have the substances subject of a monograph in the column on the left. And on the other side, we have the impurities. And they're also distributed whether or on the basis of whether or not they are used for qualitative purposes or quantitative purposes. And I will go over them one by one, starting with the reference standards for identification. So they're used for identifications of substances subject of a monograph, and they're mostly used in combination with infrared, NMR, or TLC, where the concept is really very simple. So the user needs to prepare a spectrum, for instance, based on the substance to be examined and a spectrum of the reference standard, and then can simply compare the two. So no difficult interpretation of the infrared spectrum or the NMR spectrum is needed. A simple comparison suffices. For the establishment of such reference standards, the key quality attribute is, of course, the identity. So we perform the full structural elucidation, usually by NMR and mass spectrometry, whenever this is possible, of course. But really, we use any suitable techniques that are at our disposal. We also verify the compliance with relevant requirements of the monograph, so not necessarily all the requirements, and we verify the intended use. But in general, you could say that the characterization of such a reference standard is focused more on the substance itself rather than on the determination of the impurities. Then we move over to the second category, so the substances subject of a monograph but with quantitative properties, simply called assay reference standards. They are used as quantitative benchmarks in assay procedures, mostly LC, but it can also be used in GC or even in microbiological assays. In general, we use substances which are compliant with the relevant requirements of the corresponding monograph, but this is not always the case. For instance, we might use another salt form if it's more stable or more pure. An example of that could be, for instance, pentobarbital CRS, which is used in the monograph for pentobarbital, but also in the monograph for pentobarbital sodium. We could also use another hydrate if it is more stable. And in case of really hygroscopic material, we sometimes provide the reference standard as a lyophilized substance. The content that we assign is based on the mass balance approach, which is a combination of the tests described in the pharmacopoeia, but also complementary tests that we perform. And we always estimate the uncertainty of the assigned value, and we make sure that, or we verify that this is negligible compared to the content limits of the monograph. For the establishment of such a reference standard, the key quality attributes are the identity, but of course also the content. So it has qualitative and quantitative properties. And here the characterization is so focused on the substance, but also in great detail on the determination of the impurities. So again, we verify the identity of the substance as described before, and we verify the compliance with the relevant requirements of the monograph. But we also test for volatile impurities, which could either be done by loss on drying or by a combination of a residual solvents test by Headspace GC and water determination by Karel Fischer, for instance. We verify the presence of inorganic impurities, mostly by a screening with sulfated ash, and we could also perform further testing further testing if required. We verify the homogeneity of the substance, usually by loss on drying or water, but also by residual solvents in specific cases where this could be pertinent. We confirm the assigned content or purity of the material by orthogonal methods, usually quantitative NMR or elemental analysis, but this could also be a titration or an LC assay against the previous CRS, whenever this is possible. And for parameters with a significant contribution to the assigned content, we organize an interlaboratory study. This is a lot of theory, so let's put it into practice. The example I've chosen is pemetrexate disodium heptahydrate CRS, which is used in the assay of the corresponding monograph. And this is the testing we have done at the EDQM laboratory. We performed infrared mass spectrometry identification of sodium ions and NMR. So this is clearly the identification section. Then we have verified the relevant requirements of the monograph, the LC test for enantiomer purity, the related substances test, the water test, and the assay. And some of these tests are of course also used for the mass balance approach, as I will explain in a minute. And we performed the analysis of volatile materials by a combination of water test and residual solvents. And we verify the assigned content, which I will show in a minute, by assay against the previous material and quantitative NMR and elemental analysis. Now, of course, in a heptahydrate, there's going to be a lot of water in. So for this parameter, we organized a collaborative trial, including 5 laboratories, of which the EDQM lab is one. So we send out the samples and we received results back. And if the acceptance criteria are fulfilled, then we take into account the result, calculate the mean, and use this for the mass balance approach. So this is the formula that we applied. So we take into account the water content, the presence of, or in this case, the absence, but the inorganic impurities, if there would have been any, residual solvents, and of course the LC, the impurities determined by LC, and we obtain a content of 78.5%. Now looking back to the LC assay and the quantitative NMR results, so you can see that the results obtained confirm the assigned content. Then we move over to the substances which are not subject of a monograph, so the impurities, starting with those for qualitative purposes. And they're mostly used in the chromatographic separation techniques, LC-GC or TLC. And they can be used for 2 very distinct purposes. On the one hand, we have batch testing, and then they are mostly used for identification of signals. Because imagine if impurity A, for instance, has a specified content limit, then of course we need to provide a way to identify impurity A in the chromatogram. Or for instance, this impurity could have a correction factor assigned to it. And again, we would need to be able to identify the impurity. On the other hand, we have the system suitability testing, which is done as a prerequisite to the batch testing, of course. And even though these functions are quite different, the establishment of such reference standards has a lot of similarities, and also a lot of CRS are used for both. So therefore, in this presentation, they are discussed together. For the impurities, in contrast to the substance subject of a monograph, we have a much more liberty in developing the reference standard strategy. In this case, for instance, we have a choice between providing single substance reference standards or mixtures, and I will show an example of both. So the 2 examples I've chosen, so in both examples, the standards are used for system suitability testing, specifically a peak to value ratio between 2 peaks. In the first example, we have single substances, so we have glipizide impurity A CRS and glipizide impurity T CRS. The advantage of this strategy is that in general, these single substances are much more stable, and also their concentration is easily controlled. So during the development of the monograph, we are free to choose which concentrations we need or we want, or which would be optimal, and we can simply describe that. This is in contrast to the mixtures that I will show in a minute. And also in general, we have a very good batch-to-batch continuity, because imagine impurity D is 100% pure, and impurity DCRS1 is 100% pure, and impurity DCRS2 is only 95% pure, then this would only have a very limited content on the very limited impact on the outcome of the system suitability criterion. So this is of course an advantage. The disadvantages are that this might result in a high number of reference standards, which of course comes at a cost for the user, and not always are these materials available on the market in sufficient quantities for us to establish a reference standard. Risperidone for system suitability CRS is an example of a mixture reference standard. So you can see straight away that this is more representative of what the user will observe for his substance to be examined. The disadvantages of this approach is that the batch continuity may be troublesome. Imagine, for instance, in this case, that impurity D is present at 0.10%, and that we need to establish a batch 2 of this material. It's not easy to get a material with exactly the same amount, so it might be that in the next batch, impurity D is present only at 0.05% or at 0.2%, and you can imagine that this will have a dramatic effect on the outcome of the system suitability criterion. But definitely this is something we take into account when establishing these reference standards. A second potential drawback could be the fact that since often this the concentration of the material or of this solution is the same as a test solution, very often the main peak is saturated, which would impede a proper calculation of the resolution if this would be the criterion that was described. For the identification of impurities or for the system suitability testing, we do not necessarily need to have a CRS. We can also have commercial reagents if they are available, or in situ degradation if this could be applicable. In general, we use for the strategy batches containing the impurities if these impurities are specified, because then normally batches are available on the market containing all the necessary impurities, but this is not always the case. And in case of mixture reference standards, we often provide a chromatogram in the leaflet, which helps the user to identify the peaks. The establishment of both types, so the single substances or the mixtures, are quite different, so I'll discuss them separately. For the single substances, the key quality attribute is of course the identity, because they only have a qualitative purpose. And therefore, again, we would verify the identity by any means possible. And we verify the intended use, because even if, for instance, we use an impurity A reference standard, and this would be 95% pure, but imagine that the impurity present at 5% really over-responds in the LC method, then the identification of the impurity may not be straightforward. So this is also something we take into account during the establishment. But in general, it's safe to say that the characterization of such materials is less elaborated than the reference standards that are used quantitatively, as I will discuss in a minute. For the mixture reference standards, the key quality attributes are the identity of the impurities, obviously, their content, and also the fitness for purpose. And by that, I mean, even if we have a material in which all the peaks are there, or all the necessary impurities are present, but we also have a lot of other peaks appearing in the chromatogram, which might hamper the identification, then this could be a problem as well. We verify the identity of the impurity peaks, preferably by spiking with authentic samples if they are available, or otherwise by LCMS. We check the homogeneity of the material, and we verify the intended use. And then finally, we arrive at the fourth category, which are the impurities used for quantitative purposes, so the external standards basically. They are mostly used in the chromatographic methods, LC and GC, or also TLC, and they are external standards for impurities with a response that is very different from the substance subject of a monograph. Because if not, the primary option would be to identify or to quantitate these impurities against a dilution of the test solution. If the response of the impurity compared to the main substance is only slightly different, also a correction factor could be given. For the establishment of this type of reference standard, the content of the candidate material is critical. Is it above 95% or not? Because as described in chapter 5.12, If the content is above 95%, we do not assign a content, and the user can simply assume the content to be 100%, because the mistake that is made by applying this assumption is irrelevant compared to the measurement uncertainty of the method. For this type of reference standards, we only use single substances. We don't have mixtures with an assigned content. And the main problem here is that we often use materials that are obtained via processes that do not guarantee the required degree of purity and homogeneity, because as you can imagine, it's quite easy to buy the APIs. Buying the impurities is not always straightforward. And then finally, we need to think about which salt form we want to use, because it might have an impact on the use. For instance, we take into account which salt form is easier to handle. For instance, free bases do have a tendency to be more viscous or oily substances compared to their salts, but they are also less hygroscopic, so we need to find a balance there. Also, we take into account the volatility of the material, and of course, in the end, we can't simply use any salt that we want, because we of course need to be able to procure it on the market. The salt form may have an impact on the solubility, which is an important factor to keep in mind, and it may also trigger the need for a stoichiometric conversion factor, which is something I will explain in more detail. If you have a batch of an API and this batch is present as a hydrochloride, it's safe to assume that the impurities will also be present as hydrochloride salts. And therefore, the specification limits in the monograph for this impurity are set as if they were in the hydrochloride form. Therefore, during the establishment of our reference standard, we need to identify the presence and the identity of a counterion. And if it is different from the monograph form, we introduce a stoichiometric conversion factor to eliminate the impact the salt form has on the quantification of the impurity. A big exception to this is, of course, if the impurity cannot form the salt, then it will probably not be present as a hydrochloride salt, and then we don't need to introduce a stoichiometric conversion factor. For the establishment, again, the key quality attributes are the identity and the content. So again, we verify the identity of the material itself, but also of the counterion, either by a specific test if we know what we have, or by a screening method. We determine the amount of related substances by the method of the intended use, mostly LC and GC. And we determine the volatile impurities as I discussed before. The presence of inorganic impurities is verified by sulfated ash if the amount of candidate material allows. We also have a micro ash test, which is it's an adaptation of the total ash test, but of course with lower quantities that we can apply. Or we have a screening method by LC-CAT that we can use. We perform quantitative NMR. We verify the homogeneity of the material by any suitable method. And we assign a content, of course, only if the content is below 95%, either by mass balance or by the quantitative NMR result. But in any way, we try to use the other one as a verification. So if we assign by quantitative NMR, we use the mass balance result as an orthogonal method to confirm the amount, the content. An example of such an establishment is the phenobarbital impurity ACRS. So the first, part of the analytical results came in. So the identity was confirmed. The loss on drying result was 0.1%, and the LC purity was 99.7%. So the mass balance, you would imagine, is 99.6%, and there is no need for an assigned content. However, when we received the quantitative NMR result expressed as such, so as the free base, then we obtained a value of only 79%. And also the elemental analysis did not match the theoretical composition. So we launched an investigation, and tested the presence of a couple of ions according to the chapter 2.3.1 of the Pharmacopoeia, and eventually found that there was nitrate present, which was not stated on a certificate of analysis. So this refers back to my comment earlier that we are not always able to find material which is properly characterized by the supplier. We then quantified the amount of nitrate ion by ion exchange chromatography, which was 20.6%, which corresponds to this being present as the counter ion. So in the end, we did not assign a content, but we assigned a stoichiometric conversion factor. Right, now we come to the third part, the reference standards for general chapters. Some general chapters describe analytical methods that are applied in a general sense in the European Pharmacopoeia, and some CRS are intended to be used in these general analytical methods. The aim of this part of the presentation is not necessarily to provide you with an exhaustive overview of all the reference standards we have, but it's just meant to give you a taste of what is available and how they should be used. Starting, the first examples concern CRS that are used in limit tests. The method described in the chapter for residual solvents can be used as a limit test for class A and class 2 residual solvents. And in this procedure, a reference solution is to be prepared using the class 1 residual solvent CRS, which contains ppm level of these impurities, or of these solvents, sorry. And this is used for the system suitability verification to ensure sufficient sensitivity, but also to help the user identify the peaks. Information on how to use the CRS is provided in the general chapter. So this is the chromatogram you would obtain, and this CRS is delivered with a chromatogram in the leaflet to assist users in the peak assignment. The general chapter for control of N-nitrosamines describe several methods that are describe several methods that can be used to verify. Sorry, I lost my trace. So this chapter describes several methods to analyze the several nitrosamines that I described here. And it describes also 7 CRS that can be used to verify the system suitability, but these CRS also serve as external standards in the limit tests. And again, in this case, the information on how to use a CRS is available in the general chapter. So the CRS can be ordered in our catalog. A final example is the general chapter for the determination of elemental impurities. In this chapter, no analytical method is given, but instead it describes some general validation requirements that include, amongst others, a verification of the accuracy. It is suggested to use the CRS for this purpose, but it's only a suggestion. Other suitable CRMs can be used as well. Meanwhile, we have established reference standard for 7 different potential sources of elemental composition. And it is worth noting that these CRS are traceable to the SI, and therefore they enable a metrologically sound determination, which is an element that is sometimes lacking in other reference materials available on the market. The leaflet provided with the reference standard contains the assigned content of the assigned content of the concentration of the solution, along with its expanded uncertainty and a statement on the traceability to the SI units. So it's basically like a CRM. No reference procedure is described in the general chapter, nor in the leaflet of the reference standard, and this means that the CRS can be used in principle in any procedure. It's important to note that the acceptance criterion is set by the user depending on the intended use of the method, and this is something that I will further elaborate in the coming slides. Now we move over to the CRS for equipment qualification. And as you are all aware, there is always a certain degree of dispersion in analytical results. And if you want to evaluate the conformity of a certain product, it is essential to reduce the dispersion as much as possible and to ensure that measurement capability is appropriate. As illustrated in this slide, there is an unbreakable link between the specifications of a substance, the performance of the testing method, the performance of the analytical equipment, and the reference material that is used to assess the equipment performance. Let me give you an example of that. It does not make sense to set specifications to the 10th decimal figure if your instrument only allows to determine until the 3rd decimal figure. Also, if you know that the uncertainty of the reference material, the uncertainty associated with the reference material is 2%, then it does not make sense to set a specification for your equipment performance to plus or minus 1%. So just to show you that all these parameters are tied together and that changes in one will have an impact on all the others. We can also look at it by measurement uncertainty. So if we start for a specification range of a certain product, the uncertainty of the measurement method can only comprise a part of that range, because of course you need to leave some margin for the variability of the product. And the same principle basically trickles down to the measurement equipment, which cannot comprise all the uncertainty of the measurement method. And finally, the reference material used for equipment qualification should have the smallest possible uncertainty assigned to its content. Let me show you some examples of these, starting with paracetamol for equipment qualification CRS, which is used in the chapter for Raman spectroscopy. So it is used to verify the wave number scale of the instrument, and the CRS corresponds to a material that was thoroughly characterized for identity, purity, and homogeneity, and the acceptance criteria, which are basically tolerances for the wave number, when you measure the CRS, are given in the general chapter. So it suffices to simply measure our reference standard and to check compliance to the tolerances given in the chapter. Another case is nicotinic acid for equipment qualification, CRS, which is used in the chapter for absorption spectrophotometry, UV, and visible for UV and visible light, where it is used to verify the absorbance accuracy of the instrument. Here, again, the procedure and the acceptance criteria are defined in the chapter, so it is quite straightforward to use our reference material. And the criteria given refer to the certified absorbance of the CRS. And these you can find in the leaflet. So values are given for 2 wavelengths, as well as the uncertainty given, as it is a good practice for reference materials. However, these are not necessarily required for the use because, as I said before, you simply compare to the limits given in the general chapter. We also have calcium oxalate monohydrate CRS, which is used for the calibration of the electrobalance of TGA instruments in accordance with the general chapter, with the corresponding general chapter. And here it is important to note that no testing procedure nor acceptance criteria are provided in this general chapter, but they are provided in the leaflet of the CRS, along with a certified mass if this procedure is applied and the uncertainty assigned to the content. Important to note that in this case, it's up to the user to define appropriate acceptance criteria, which, as I explained earlier, depends on the performance required in your laboratory, which is linked to the product specification of the product that you want to test. And the final example I have is sodium aminosalicylate dihydrate for equipment qualification, which is used, amongst others, in the chapters for semi-micro determination to qualify the instrument. So to establish this reference standard, we first of all verified the compliance with the requirements of the monograph. We performed an inter-laboratory study for the content assignment, and we, of course, verified the homogeneity on a large number of samples. And finally, calculated the assigned property value as a mean result of the inter-laboratory study. We also provide the assigned uncertainty, which is basically calculated using the uncertainty coming from the inter-laboratory study, combined with the uncertainty coming from the homogeneity study, multiplied by a coverage factor of 2 to obtain a 95% confidence level. The test procedure, certified value, and uncertainty are stated in the leaflet of the reference standard, but it is up to the user to define suitable acceptance criteria. Because again, as I explained earlier, this depends on the methods you want to use and the materials you want to test. However, this was proven not to be straightforward because we have received many help desk questions on the subject, and therefore in the leaflet we include suggested acceptance criteria. It is important to note that, of course, other criteria may be chosen, and you still need to demonstrate the suitability to the competent authorities, even if you use the suggested acceptance criteria. As a little sidestep, these reference materials can also be used for another purpose. Taking an example that I actually took from our lab. So we used it to construct a meteorological control chart by repeatedly measuring the CRS over a time period. You can see, for instance, that we have at one point above the action limit a couple of years ago, which of course we could then investigate. And based on this meteorological control chart, we estimated systemic bias compared to the assigned value of the CRS, and also the uncertainty, which is due to the precision of our instrument, or more or better, the lack of precision. With this, we have calculated the overall measurement uncertainty of our method, which is asked by some competent authorities. And then finally, I added these slides to the presentation because it's the most common question that I receive. Can we use the European Pharmacopoeia primary reference standards to establish a secondary standard? First, I added an extract from the EU guideline for GMP, which states that you should preferably use the primary reference standards, but it does leave the door open so you can use secondary standards on the important condition that the traceability to primary standards has been demonstrated and is documented. So basically, yes, it is allowed. It is definitely not the intended use of our CRS, but it is possible. But it is important to note that this is fully under the responsibility of the user. And obviously, this is only possible for the same property that is used for which the CRS was established. As I said before, it is necessary to ensure that methodological traceability to the primary standards, which means that in practice, it's only feasible for qualitative properties, which I will explain with a theoretical example. Imagine we have a reference standard used for identification by IR. So the intended use of the primary is, of course, identification. And the intended use of the secondary standard should also be identification. So an assay is not possible. So the intended use of the reference standard is simply to measure, to record an infrared spectrum of our reference standard and compare it to the spectrum of your substance to be examined. And then if the comparison complies, then the identity can be proven. It is possible to either establish an in-house secondary standard, or to simply store the reference spectrum in your infrared equipment, and then use this stored spectrum to compare to the spectrum of the substance to be examined. And this is because the identity is basically a binary decision. It's a yes or no, and there is no propagation of the uncertainty. If the spectrum you obtain for your secondary is identical to the spectrum you obtain for the primary, then it doesn't matter whether you compare to the spectrum of the primary or the secondary. This is very different for reference standards used for assay. So here the intended use of the primary is assay, and of course of the secondary it's probably assay as well. So the intended use is clear. And now the question is, can you establish a secondary in-house reference standard? And the answer is no, you can't, because you can't propagate the uncertainty. To assign the uncertainty to your secondary reference standards, you would need, amongst others, the uncertainty which is associated to the primary European Pharmacopoeia standard. And this is not given because it's negligible to the if you use it for the intended purpose. So based on that, you can't use your secondary for the for an assay method. This is a very brief explanation on the subject, and I would like to highlight that last year a presentation was given by the USP and EDQM on this topic. So the link is given here in the slide, and this presentation is still available on our website, and I encourage you to consult it if you have doubts on the subject. So allow me to draw some conclusions from the presentation. The establishment of our reference standards is adapted to the intended use according to the key quality attributes, and the suitability for any off-label use should be demonstrated by the user. Reference standards described in the general methods are highly relevant tools to ensure reliability of your measurement results. And the reference standards for equipment qualification are specifically characterized specimens that may be employed also for other purposes. And yes, you can establish secondary standards, but not for all uses. With this, I would like to thank you for your attention, and I hand over the floor to my colleagues.
Oshin, sorry, we can't hear you. You need to unmute.
Sorry about that. Thank you. Okay, so hello everyone. My name is Oshin Daly, and you're all very welcome to this webinar. So I'm a GMP inspector here in the Certification of Substances department in EDQM, and my presentation is just about what GMP inspectors expect on reference standards. So essentially, during our inspections, we check for compliance with European regulations. And for reference standards, this involves both EU GMP Part 2 or ICHQ7, and both of those are essentially the same guidelines. And there are also requirements outlined in the European Pharmacopoeia, specifically section 5.12. And I also just want to kind of clarify at this point, there are, of course, further requirements outlined in EU GMP Part 1 in Chapter 6 for quality control, but given EDQM inspections only relate to APIs, I'm just going to talk about Part 2 today. Okay, so in EU GMP Part 2 or ICHQ7, Section 11 for laboratory controls describes the requirements for reference standards. So paragraph 11.17 talks about obtaining primary reference standards, documenting the source, maintaining records for their storage, and to use in accordance with the supplier's recommendations. And it also clarifies that you can use the primary reference standards without testing if they're obtained from an officially recognized source and when stored under the appropriate storage conditions. So how to comply with these requirements then? So firstly, it's obviously important to obtain the correct reference standards. So for our inspections, this would mean obtaining the European Pharmacopoeia standards from the EDQM. And you can do this quite easily by accessing the EDQM catalog via this link here on the screen. So we can see here an example of the standards available for ibuprofen. And just important to note, you may need multiple standards, not only the primary CRS. So for example, if you're carrying out testing for related substances in line with the monograph, there are multiple other standards for impurities and for peak identification required as well. So this is something we would check during inspection, whether all of the required standards are available, whether they're being used, and whether the testing is being carried out in line with the monograph. Okay, the next thing we would check then is whether the reference standards are being stored and used in accordance with the instructions on the information leaflet. This here is an example of the leaflet for ibuprofen, I believe. And I've just highlighted here that, you know, once the container has been opened, its entire content must be used immediately. And this is a common issue we see on inspections, whereby the primary reference standard is essentially being used as a multiple use standard, which is not in fact in line with the instructions for use. Also important to consider the storage conditions. So most of the EDQM reference standards require storage conditions between 2 and 8 degrees. But of course, it's important to check this specifically for the standard being used. And we would follow up on this during the inspection. You know, we would check the data for the fridge, for example, and perhaps the qualification of it as well, if we have time. And then lastly, very important to check that the standard batch is valid at the time of use. So again, this can be done quite easily by accessing the EDQM catalog with, on the previous slide, and you can download the batch validity statement from there. Okay, the next part of EU GMP Part 2 or ICHQ7 is paragraph 11.18. So this really describes the requirements for establishing primary reference standards, and this mostly concerns cases where there is no official standard available. So for example, if it's a non-pharmacopoeial substance. So it talks about doing appropriate testing, you know, having the appropriate documentation, and there is a lot more detail, in fact, in section 5.12 of the Pharmacopoeia. So this would be the guidance document that we check compliance against if we come across this situation. But to be honest, for EDQM inspections, we don't see this practice that much because our inspections are carried out in the context of the CEP procedure. So there must be a monograph to have a CEP, of course. So in theory, there would always be an EDQM reference standard available for the substance within scope of our inspection. Okay, then the next section, 11.19, talks about secondary reference standards. So again, the requirements here are that they should be appropriately prepared, identified, tested, approved, and stored. And the suitability of each batch of secondary reference standard should be determined prior to use by comparing against a primary reference standard. And also each batch of secondary reference standard must be periodically requalified in accordance with a written protocol. So how to comply with these requirements then? So firstly, it's very important to document and justify the approach if you're going to qualify a secondary standard. So there should be a written protocol documented in advance of the exercise with the acceptance criteria defined. The primary standard should be traceable and be used correctly, as I previously mentioned. So this would be in accordance with the information leaflet and the instructions for use. So again, we're looking at batch validity, storage conditions, and we would check if it's the first time the vial is being opened. Then for the secondary standard, as my colleague mentioned, of course, it should be used for the same purpose as the primary standard, and there must be a consideration and justification for the shelf life or the retest date. So as mentioned, there must be a periodic requalification. The storage conditions must be justified, and whether it's a single-use or multi-use standard. And again, section 5.12 of the Pharmacopoeia contains some further guidance on secondary standards. So in summary then, what we would look at during inspections is whether the current European Pharmacopoeia reference standard is available, whether it's being used correctly in accordance with the method in the monograph or any other registered method. If the storage conditions, labeling and compliance with the instructions for use are okay, and then we would also look at the approach to secondary standards if the company is qualifying those. So again, we would check the qualification against the primary standard, the shelf life, storage conditions, labeling, and the all-important data to support the approach being used. So just to give you some information on some of the findings from our inspections, so I conducted a review of almost 200 EDQM inspection reports going all the way back to 2011. So we found 50 deficiencies concerning reference standards. 5 of these were classified as major and 45 as other deficiencies. However, it was noted the incidence of reference standard deficiencies is decreasing. So I suppose in the first few years of this review, there was deficiencies identified in approximately 44% of inspections. But in the more recent inspections, the incidence of these types of deficiencies has decreased. So it's currently at about 14% of our inspections, we would find a deficiency on this topic. And also just to mention, we do find, if there are re-inspections conducted, the problems do tend to be addressed by the companies, which would indicate, effective CAPAs being implemented to address our previous findings. So just to finish with the presentation then, I'll just run through some example deficiencies that we find during our inspections. So firstly, concerning the primary reference standards from the Pharmacopoeia, so we might find that they're simply not available at the site. So they might be using some other reference standard or their own standard which hasn't been appropriately qualified. Another example, we found a company assumed that the content of the European Pharmacopoeia standard was exactly 100 milligram, which was not correct. So this resulted in the results for release batches not being accurate. And then another example, we found that a company was using a diluted sample instead of the European Pharmacopoeia related substance reference standard, which was required by the test method. So in this case, instead of using that, they just diluted the sample solution 1000-fold, which is clearly not justified. Okay, so then in terms of deficiencies for in-house primary reference standards, So, as I mentioned, it's not that common that we would see this approach, but just an example of an issue where we did come across it. So, the first point here, appropriate testing was not performed to fully establish the identity and purity of the primary reference standard in the absence of an officially recognized source. So, in this case, I guess the company simply just didn't follow the requirements outlined in section 5.12 of the Pharmacopoeia. The second example then, only the first batch of internally manufactured reference standard was established as an in-house primary reference standard with a full physiochemical characterization. And this was used to qualify a batch of working standard. However, after that, the suitability of the working standard batches was only verified against the previous working standard and not against the fully characterized primary reference standard. So for each batch of working standard or secondary standard, we would expect that it's qualified against the primary reference standard and not another secondary standard. Okay, so then in terms of secondary reference standards, some more examples of deficiencies. So the first one here, only internally manufactured secondary standards were used. However, they were not qualified against European Pharmacopoeia primary standards. And then second point, the suitability of secondary standards was not adequately determined by comparison against the CRS, so only an identification test by IR was performed. So of course, if the standard is to be used for quantitative purposes, it's inappropriate just to use an identification test. Then in terms of the requalification period, we have an example here. There was no evidence that secondary reference standards were appropriately requalified. So the SOP outlined an expiry period of 1 year. However, there was no stability data or other documentation to support such an expiry date. And then the last example, there was a lack of traceability in relation to the qualification of secondary standards. So the secondary standard COA did not include reference to the batch number of the official European Pharmacopoeia primary standard against which it had been qualified. Then in terms of storage of reference standards, as I mentioned, we would check whether they're stored in accordance with the instructions for use. So in a lot of cases, we find that in fact, reference standards, the primary reference standards are not being stored in the fridge, but they're being stored at room temperature. And then in some other cases, we might find that there's no temperature monitoring of the fridge being used to store the reference standard. Then in terms of the batch validity statements, this is another relatively common deficiency whereby the batch validity statements are not being checked to ensure that the reference standards being used are valid at the time of use. So I think that's it from me. So thank you very much.
Good morning, everyone. Welcome again to today's webinar on reference standards. My name is Marine Clausel, and I'm heading the operations division within the supply chain department. The presentation will deal with handling, dispatch, and the practicalities of reference standards. And for this, I'll be guiding you through several key topics. So we'll start with the ordering process, then with an explanation of what labeling means for the EDQM and its reference standards. We'll also discuss what is expected from you as a user, and also how to prove the validity of the reference standards you are using. We'll then briefly touch with shipping and storage conditions. We will go some safety features related to the reference standards, and conclude with other sources of information and frequently asked questions. So let's start with the ordering process. So for the ordering, before ordering, your primary source of information is the online catalog, where you have the link here displayed on the slide. This link brings you to the EDQM store. And when following that link, besides the European Pharmacopoeia Reference Standards catalog, please note that there are others catalogs specific to the WHO standards, one for the International Standards for Antibiotics, the ISA, and another one for the International Chemical Reference Standards, the ICRS. There is a more direct link to the European Pharmacopoeia Reference Standards catalog, which is provided here. This online catalog is only available in English and is updated on a daily basis. So when you are searching for a reference standard, you have different ways to search the reference. It can be by substance name, by catalog code, by monograph number, CAS number, and SDS product code. Regarding the CAS number, please note that the CAS numbers are not independently verified by the EDQM and that they are here for informational purposes only. You can also here download the full catalog, either as a PDF file or as an XML file, and you can, if you wish so, integrate that within your IT system. And we know that some users do interface that with their LIMS. So here is an example on how you can prepare for customs and reception in your laboratory. So this is the case for the coding for system suitability reference standards. It's a controlled substance. And in the past, obtaining a permit for controlled substances from the French authorities took between 8 and 10 weeks. We now have a new agreement which allowed to reduce the time to 2 to 3 weeks. We have also included in the additional information section the actual composition of our reference standards to help you with your permit requests. You will find on this page the direct links to the safety data sheets and the origin of goods document. For the origin of goods, we now also include the country of origin to meet some regulatory requirements in some countries. We have also a section with the availability and if we have sales restriction on that reference or not. For certain standards, specific documents are required. For example, for precursor, psychotropic, narcotic substances, an original import permit is necessary and the authorities won't accept web copies. We have 2 items in the catalog classified as ODS, or the ozone depleting substance, and for them you'll need a valid lab ODS number or a license depending on your location. Be cautious here because some countries may refuse the import of these substances. For biologic reference, for biological materials, especially in the US and in Australia, please ensure you have the correct import permits as regulations are becoming more stricter. And you can find other documents from the website. First one is a letter replacing the certificate of analysis. And the second one is a legal framework and diplomatic status of reference standards for customs purposes. Now that the order has been prepared, let's see how we can do the order processing itself. You can order directly from the EDQM or from resellers. However, if you're not purchasing directly from the EDQM, be wary of counterfeit products and some misleading information, such here the example with a fake expiry date. We have also seen some cases where some falsely claimed assigned values have been provided. We do not have official distributors, but we provide a list of companies that resell our products, which can be useful in countries where the import processes are complex. We also offer on our website a tutorial video on how to use the web store. And also, please also be informed that the reference spectra are now emailed as PDFs. We are not providing paper copies anymore. As already said, we have recently revised our certificates of origin. They are now available online from the catalog. But be aware that the EDQM cannot issue a R1 movement certificate, which some importers use for reduced tax at import. Once you have placed an order, you have 24 hours to change it or to cancel it. After that, it's locked for preparation and you will receive an acknowledgement of receipt. You have an example here on the slide, and please check it carefully. We still prioritize door-to-door shipment. We also split deliveries according to shipping routes and book flights when we are preparing the parcels. Due to some ongoing restrictions, especially for ice shipment, it's not always possible to guarantee door-to-door delivery, and then it will be delivered to the closest customs airport. Ensure your broker is proactive in handling customs issues to avoid delays. The COVID pandemic has affected shipment, and shipping issues do continue with last-minute flight cancellations and some changes, or some countries blocking parcels for sanitary reasons. We continue to prioritize door-to-door shipment where possible, but we cannot guarantee that we will be always successful. Now let's discuss the labeling at the EDQM. So for the EDQM, the label includes the primary label, the pictogram, the secondary label, displaying also some pictograms, and the leaflet. All these together is the label. So on the primary label, that's the one which is on the vial, you will find the information such as the catalog code as it appears in the catalog, the batch number, and the sub-batches, the approx quantity per vial. It's a peel-off label as well. You have a 2D barcode. You have the reference standard name in English as it appears in the European Pharmacopoeia text, and some sentences for customs. I'm pleased to tell you today that a new label is on the way that will display additional information. So the 2D barcode will be replaced by a QR code, which is able to embark more information. We will still have the RS name as it appears in the pharmacopoeia text, but we will be adding the reference standard component. And for mixture RS, the main components contributing to the safety risk are disclosed. We are also adding the cast numbers, and again, they are not independently verified by the EDQM. So the deployment of this new label is gradual, and both labels will exist in parallel for a certain time. The safety information, including the pictograms, is provided both on the primary label as a tag in the current label and on the package, the external package. We are currently transitioning on the primary label to a smaller integrated pictogram, which is directly to be integrated in the new label to come. So when there is a safety risk for our given reference standards, the tag in a new label will disappear and the pictograms will directly be integrated within the label. The secondary package also includes pictograms and warning statements in the language of the destination country. If you notice any error, please let us know so that we can correct them. And the leaflet provides additional information such as the assigned content, the content in milligram or milliliters of the chemical entity in the container, some assigned potency, for instance, for biological substances, and also some information on the storage conditions. This information are not mentioned on the primary label or in the catalog, so be sure to read it carefully. And the leaflet may also include chromatograms, some conversion factors, and other critical information, especially for controlled substances. So always refer to the leaflet for specific instructions and storage conditions. So let's move on what is expected from you as a user. So before using a reference standard, you should check that the batch number is valid for your analysis at the date of your analysis by printing the batch validity statement, the BVS, from the online catalog. You also need to inspect the container and the closure system for integrity, and you need to ensure the reference standard has been stored properly at your facility. You also need to allow the reference standard to equilibrate to the lab temperature before opening it. That's to avoid condensation issues, and this is especially important in humid climates. You also need to use the standard as is, meaning without desiccating it or without drying it, unless it's specifically written in the leaflet. And we provide different types of vial. We have a V vial for filling weights below 15 milligrams to ease the recovery of the powder. And for filling weights above 50 milligrams, we have standard brown glass vials, which are also used for evaporation and freeze-dried references. So how can you prove the validity of the reference standards you are using? We do not provide an expiry date or a retest date. We ask you to use the batch validity statement, the BVS. And to get the BVS, you have to connect to the online CRS catalog and to follow the dedicated link to the BVS. In this example, we are using ibuprofen impurity B CRS. So you need to ensure that the batch is currently valid by printing the BVS on the exact date of your analysis. Here is batch 11 we are talking about. So the BVS will show that the batch was official and valid at the time of printing, including all the sub-batches. This is crucial for inspections, as said by my colleague, to prove that you used the correct reference standard. And please also note that multiple batches can be valid at the same time. For instance, we have here batch 10 and batch 11 that are valid, so ensure you are printing the correct one for your analysis. When a batch number is running out of stock, we typically allow a 6 months transition to the next batch, and this rule applies unless there is a quality issue. And after printing the BVS, you can attach the peel of primary label to the certificate to prove you used the official CRS batch at the time of your analysis. Now let's discuss shipment and storage. So our packing material is designed to prevent damage, to maintain the temperature, and to comply with all the applicable transport regulations. And the packaging also ensures that the prescribed temperature is maintained for at least 3 days. For instance, for a plus 5 degrees shipment, we allow a range between 1 and 8 degrees C. The dispatch conditions are selected based on the reference standards needs, so short excursions from the recommended storage temperature during shipment should not impact the quality, but this is assessed on a case-by-case basis. The EDQM storage conditions are established for long-term storage. They are based on stability data, the data we have on the bulk material, some monitoring data, literature data, and any other information we may have. You may store reference standards differently in your lab, but you need to ensure that you can justify these conditions during inspections. It's your responsibility to demonstrate that. And at the EDQM, we typically store at one level below the monograph's recommended temperature to ensure the optimal stability. For instance, if the monograph states that the storage temperature is room temperature, we do store at +5 degrees C. If it's between +2 and +8 degrees, we store at -20 degrees if relevant, and so on. Any change in the shipping or the storage conditions are communicated through the RS News, which is published monthly on our website, and we aim to inform the users at least 1 month in advance before any change takes effect. Now let's talk about safety of the reference standards. So for substances of very high concern, it can be, for instance, anti-cancer drugs, we clean the external part of the vials after the manufacturing to protect the users. However, always wear gloves when handling the vials, especially after removing the plastic cap. The safety information is available through the safety data sheets for the chemicals and the safety data statement for the biologicals. These documents do exist only if an hazard has been identified. For references where no hazard has been identified, there is no such document. These documents are to be found on the website in the corresponding catalog page, and they are provided for occupational health and safety, and they are not part of the quality standard. Finally, let's cover some other sources of information and the frequently asked questions. So you can find a wealth of information on our WebMPED website, including the subscription option for the EDQM newsletter and the RS News. As already said, the RS News is published monthly and includes updates on the storage, the shipping, and other critical information such as sales unit change, price change, quantity changes, and so on. Something which is quite new is that you can now subscribe to the EDQM RS newsletter to get it directly into your mailbox. So you have the link provided here on the slide. Do not hesitate to register to that newsletter. And now I've also prepared a compilation of the top 10 frequently asked questions we received at the EDQM. Sorry. So the first question is, how can I obtain the certificate of analysis? So that's a very common question, and the EDQM does not provide a certificate of analysis. All the necessary information is included in the leaflet. And if you need to justify that to the authorities, we provide a document on our website explaining why we don't issue certificate of analysis. The second question is, I do not find the expiry date. It's your responsibility to prove you used the official batch at the time of testing using the BVS. We do not provide any expiry date nor a retest date. Another important point is that once the container has been opened, the weighing should be carried out immediately, and any further storage or reused is not guaranteed. The third question is regarding the purity. I do not find the purity. If it's not mentioned in the leaflet, it means that it's not required for the monograph test and for the RS intended use. As already said by my colleague, don't assume a 100 purity test, unless it's specified in the leaflet. If there are two weights declared, one on the label and one on the leaflet, which one should I use? We often see confusion over declared weights. So the weight on the label is for commercial and customs purposes, while the leaflet provides the information for the analytical analysis. So when doing your analysis, use the information on the leaflet. I cannot recover the quantity from the vial. Sorry. So if you cannot recover the quantity from a vial, it may be to the small amount of substance in the vial or some handling issues. So you can gently tap the vial and use an anti-magnetic device to collect the powder. You can also allow the sample to equilibrate to room temperature before use. And another option could be to use differential weighing. The parcel arrived with cold pack stored. Is it still suitable for use? May I obtain stability data? If your parcel arrived with stored cold packs, it's usually still suitable for use. If you're concerned, contact our help desk and we'll assess the situation. Regarding the stability data, we do not share stability data. Is there a QMS applied to ERAS reference standards? May I have a copy of the certificate? So the EDQM follows the principle of ISO 9001 and ISO 17034 for the conduct of laboratory studies and the reference standard production. And we also have the certification ISO 17025 for certain techniques used in the establishment of the reference standards. The certificate is available from the website in the section EDQM About Us. May I store the RS at different conditions than those stated in the catalog? So you may store the reference standards under different conditions than those recommended in the European Pharmacopoeia, provided you can demonstrate that the integrity of the standard is maintained. Again, it's your responsibility to demonstrate that. I want an old BVS or an old leaflet. For the leaflet, we can provide it upon request. You just have to ask the question in the help desk, but we cannot provide an old BVS. That's not possible because it's valid only at the date of the printing. I disagree with the cast number provided in the catalog. So if you notice a discrepancy in a cast number, please inform us. Again, we do not independently verify the cast numbers, but will correct any mistake we identify. If you still have a question or a comment, feel free to use our help desk system, which is available on the website. We also regularly update the frequently asked questions section, but if you can't find an answer, we are here to assist. So thank you for your attention, and I think we have time to answer some questions.
All right, I will go first. So the most common question that we received is the discrepancy between something I said and something my colleague Oshin said about secondary standards. I was talking from a, let's say, analytical lab point of view about the measurement uncertainty. I have never said that it's not allowed to do that. I just said that it in practice could be difficult because we don't assign, we don't give or the we don't provide the associated uncertainty of the content in the leaflet. So therefore, you can't propagate this uncertainty into the secondary. My colleague was more speaking from the GMP's perspective, where it is stated that this is possible, but you need to be sure about the data that are generated. So in practice, I think it's difficult, but it is allowed. Let me look at some other questions. Can we get the CRS structural elucidation data from EDQM? No, we only provide data and information that is linked to the use of the reference standard. So when we provide a CRS, you can assume that we have performed sufficient structural elucidation. I see a new question popping in. Why don't we provide the uncertainty? This is because, well, as I said before, And as Marilyn said as well, we only provide data which is needed for the use of the CRS as it is intended for the as it is intended. So because the specifications set in the monograph already take into account the uncertainty of the reference standard, and we assure that the uncertainty of follow-up batches is negligible to these specifications, you don't need the uncertainty to perform the test as it is described in the monograph. Right. How are the expiry and or the retest date assigned to reference standards? What is expected to be able to assign expiry date for in-house reference standards? We don't assign an expiry date to our reference standards, but we have a two-pronged approach. On the one hand, we have a retest program. So on several points in time, we retest our CRS and verify if they're still suitable for the intended purpose. On the second hand, on the other side, we at the time of establishment, set up a stability study where we get an idea on the expected stability we can have over the coming years. What is the, sorry, what is, so on the second part of the question, what is expected to be able to assign expiry date for your in-house reference standards? Really, I cannot comment on this. This depends on your establishment of reference standards and is, of course, under your own responsibility. Is there any procedure to get the characterization data report from the CRS from EDQM? So no, we only provide data that is needed for the intended use. What is the difference, the definition of a chemical reference substance? Does it include, for instance, small molecules that are chemically synthesized? Yes, so the definition is given in 5.12. I won't repeat it here, but basically it can either be single substances, but it can also be mixtures. Regarding the nature of the substance, it can be indeed chemically synthesized compounds, but it can also be substances that are extracted from plants or antibiotics extracted from other organisms. So there's really no limit there. I have received also some questions about WHO standards and USP standards, but you will understand, of course, that I am not in a position to comment on these. If a response factor is outside the limit defined in a specific monograph and a correction factor is not provided in a monograph, then, sorry, the questions keep jumping from left to right. I need to reorganize myself, sorry. Just a second. Right. What is the acceptable variation between theoretical and experimental values in the elemental analysis? This is a very good question. Well, in fact, the elemental analysis is not performed in the EDQM lab. This is outsourced, and the lab to which we outsource provides uncertainties. So as long as the value obtained is within the uncertainty of the method, then we accept the result. What else? Yeah, so coming back to the question I was trying to answer before, if a response factor is outside the limit defined in a specific monograph and a correction factor is not provided in the monograph, then how the calculation/results can be done interpreted. This is taken care of during the elaboration of the monograph. So during the elaboration, the response is compared and a correction factor may be introduced. If no correction factor is introduced, you can simply assume it to be 1, so no need to implement a correction factor. In which cases can we use a secondary reference standard, and to what extent should this be characterized? Well, in what cases I discussed during the presentation. Also, again, I remind you about the link I used I put in my presentation, which will provide you more information regarding the second part. So what extent of testing you should perform. This is of course not for me to comment on. This is under the responsibility of the user basically. If elemental analysis results of the CRS varies with relation to the sample, but it complies with other characterization parameters, then how the results for elemental analysis can be interpreted. I'm not sure how I should interpret this question, but I assume you're talking about, for instance, if there's a certain degree of water present or a certain degree of impurities present, we recalculate the theoretical composition of the sample based on the amount of water present or the amount of related residual solvents present. So we tend to take these into account. I hope this answers the question. For a standard that has an assay and a related substance method, the assay by HPLC versus a previous batch might be 100%, but related substances result might be 99.9%. Which results, related substances or assay, would be used in the mass balance equation? Good question. Well, first of all, if these results are really so close to each other, then I wouldn't assume there's a problem because this is within the uncertainty of the method. But we always use the mass balance approach for our content assignment, because otherwise we would have a secondary standard, and we try to have primary standards as much as possible. Can response factors provided by EDQM be not accurate enough when used in lab equipment? For instance, a PDA detector used instead of a dual length detector. These correction factors provided in the monograph are only given with one decimal figure. So anyway, there is a certain lack of accuracy there. However, it is assumed that this lack of accuracy is in comparison to the uncertainty of the method is irrelevant. So also, depending on the instrument you use, this should not have a great impact on the response factor. So basically, you can just apply the correction factor given in the monograph. I would like to ask about the basis of a reference standard. Based on the reference standard certificate of analysis, it is written that the purity analyzed by LC is 100% and water 5%. Just to get clarification from EDQM, is this reference standard in as an anhydrous basis? The reference standard was bought from WHO. Yeah, so I really can't comment on that. We don't provide individual results such as the purity by LC and water testing. We only provide the content. A CRS that has no purity on the certificate, do we assume to be 100%? Marilyn also mentioned this, and I saw some questions, other questions about this. Just to be clear, what she said and what I said does not contradict each other. So you have to take into account that our CRS are established for the intended purpose only. So if no purity is stated on the certificate, can we assume 100%? No, because if the CRS is not used as an external standard, then there is no, then extensive content assignment was not done, and you should not use it as an external standard. You may, but this is based on your own responsibility. What I said about assuming the content to be 100% was only when the CRS is used as an external standard in the monograph. More information on this is given in chapter 5.12 of the Pharmacopoeia. I don't know how long I've been answering questions. I will see if there's any other questions that need to be answered. No, I think I've tackled everything. Just one more thing. If your question has not been answered or if the answer has not been clear, please put your question, submit your question to the EDQM help desk, and we will get back to you with a more lengthy response. And now, Oshin, I hand over to you.
Thank you, Bart. Okay, so the first question I see here for me, I would like to know if it is acceptable to verify the batch in use on the EDQM catalog periodically, for example, once a month instead of just before use. So I guess, you know, we would take a fairly pragmatic approach to that. So checking it once a month would seem to be a reasonable time frequency, but I guess the question would then arise if you know, you end up using something that is no longer valid, then I suppose we would look at how would this be handled. So would there be a deviation raised to check if there's any impact on using an invalid reference standard? So I guess, I mean, yeah, it's based on how you handle if that situation arises. There's another question here. What about where commercially available secondary standards traceable to EDQM are purchased and come with an appropriate COA? Is retesting required in-house or requalification? So I suppose in this case, you're essentially talking about outsourcing a GMP activity. So what we would look at there is, you know, is this outsourced activity appropriately controlled, and does the, I suppose, contract giver have appropriate oversight of that activity being conducted by another entity? So I guess we would treat it as similar to other outsourced activities. Yeah, a lot of other questions came in about you know, using secondary standards, but I think my colleague clarified that, so I don't need to answer that again. Let me just see. Okay, there's not too many other questions for me. Here's one. Why is Chapter 512 stated to be for information only when inspectors expect labs to follow it for establishing in-house reference standards? So, I mean, I guess in this case, like 5.12 for primary reference standards contains a lot of useful information and guidance. And I guess if you are not following the approach taken in Chapter 512, then I guess there will be questions raised in terms of whether there's appropriate data to support the fact that you've characterized a primary reference standard. So, I mean, I think it's pretty useful and detailed information that should be followed. That should be followed in any case, even if it is stated as for information only, because what other way could be used to characterize a primary reference standard? Okay, then the next question there's for me, which accreditations for laboratories producing or establishing in-house reference standards are needed in order to meet the requirements of regulatory authorities for pharmaceutical industry? So I guess this might be referring to other commercial laboratories referenced in the first question I answered about providing commercially available secondary standards. I'm not aware of any specific requirements for certain accreditations. I don't believe that's specified in any GMP guideline or similar. So in this case, as I mentioned, if it's up to the contract giver to ensure that the outsourced activity is appropriately controlled and managed. Just looking down through the questions, I don't see. Oh, actually, sorry, there was one about what is interpreted as immediate use of a reference standard. Is this 1 minute or 1 hour or a day? So, I mean, we wouldn't go to such detail as to be timing, you know, how long the reference standard vial has been opened in terms of minutes or hours. We would just expect that, you know, when it's opened, it shouldn't be used after that qualification exercise is complete. I guess that would be the practical interpretation of immediate use. So obviously open it when you start your the testing exercise, and then once it's no longer required, it shouldn't be used again. So I think that was all of the questions for me. Yeah, I think so. So I guess I'll hand over to Marilyn. So thank you very much.
Thank you, Wei-Ting. I think most of the questions have already been answered by my colleagues. I have a first one. What about a monograph that needs for material to be weighed out by, but it's very sticky and very small quantities present, e.g. 10 milligrams? Can we not use content of full vial without weight? If not, what do we do? So if it's not described in the monograph or in the certificate that you have to use the vial as full content as such, you can't consider that you can use the vial as such. You need to weight the exact amount needed. If you have difficulties with taking the powder out of the vial, if it's a liquid, you can centrifuge it to try to collect the amount in an easier way. If it's a powder or for any reference, please don't forget to leave it the time for the vial to come back to room temperature. You can also gently tap the vial on the bench, to try to collect the powder at the bottom of the vial. You can use an anti-magnetic device or use different weighing as already suggested. And if these advice are not enough, feel free to ask a question on the help desk for your specific for the specific reference you are working with. So we could perhaps provide more relevant information. Can I check the validity of the CRS batch by temporarily periodically downloading the CRS web catalog? No, no, you can't do that. To prove the validity of the CRS batch you are using, you need to print the BVS at the date of your analysis. That's the only way. The purity that cannot be assumed at 100% has been already discussed. Another one, is it advisable for the storage temperature to be maintained one level below the monograph's prescribed specification? At the EDQM, we are doing that to ensure the long-term stability. You can use another storage conditions provided you can prove in case of any inspections that the integrity of the reference standard is preserved. If the reference standard consists of 2 compounds, can the EDQM also support information about the composition of each compound? I'm not sure to fully understand the question. It's perhaps related to the new label where we disclose the composition of various mixtures when they contribute to the hazard classification. The information here is just for hazard classification and information purposes. If it's not needed for the test to be performed, we won't provide more information. On the validity of the CRS in an event days, there is a new batch for which is different from the one you have. Do you discard the previous batch immediately or what? No. When we are running out of stock of a batch number, we allow a 6 months transition period during which the batch is still valid. After these 6 months, you have to use the new batch number. If we have a quality issue, then it's another story, but for the normal transition, it's a 6 months period. How long should take the calibration of the recent standard to lab temperature before opening in case of refrigerator and in case of freezer temperature storage conditions? Well, it all depends on your location, the climate, and so on. Use, let's say, common standards, few hours to allow for the standard to reach just the room temperature. Avoid condensation. That's the main issue. Is there something else? Are all reference standards considered as for immediate use? Yes, they are. And I think the last one, how does EDQM ensure the availability of the CRS? We have had the experience that there are often some shortages with impact, which impact the laboratory work. Yes, in some cases we have a shortage. They are by chance rare. We also put in place, in some cases, sales restrictions to ensure that every final user has a chance to get the material to continue doing its work. But we are all at EDQM working hard to avoid such situations. I think I'm the end of the questions. Yes, there is no more question on my side. I hand over to you, Celia.
If there are no more questions, we can close the webinar. It's tell us, Bart or Oshin, any more questions to answer?
Let me have a look here.
No.
Yep, there's one here for me that came in while I was answering the other questions. Oshin, is there a difference in what is commonly done in using primary or secondary standards for batch release of final drug products? As I kind of briefly mentioned during my presentation, EDQM only conducts API inspections, so I don't think it's appropriate for me to talk about what's happening at final drug product sites of manufacture. Okay. Then there's another question. Would you accept a lab that establishes an in-house secondary reference standard and qualifies it against USP reference standard, or would you always expect to see qualification against the European Pharmacopoeia? Look, I guess our inspections are always conducted in the context of the CEP procedure, which is, which involves, testing against the European Pharmacopoeia monographs. So, yeah, if that specific standard is in the monograph, then I guess we would expect to see the European Pharmacopoeia standard being used. Yeah, so that was all the remaining ones for me. Thank you.
I found.
Okay,
sorry,
yeah, go ahead.
I found two. How do you set specification limits for stability testing? Unfortunately, we don't share this information. And the other one I found, when confirming mass balance calculation by QNMR, what would typically be deemed as an acceptable result as confirmation? Well, and the question is 1% or less. Well, this depends on the use of the CRS. For an assay reference standards, definitely below 1%. For impurities, yeah. Also, there it depends on the content. If it's anyway above 95%, then we can be a bit more lenient. But in general, the value should be below 1% difference, yes. Preferably well below 1%. but I can't share a hard limit.
Okay, so if there are no more questions, we will close our webinar for today. And of course, you are very welcome to ask more questions on our help desk. The link is on your screen, and we will send you also the presentation in PDF format and the recording in the next few days for you to have a look again. And thank you for answering our short feedback survey when you exit this webinar. So thank you again, and have a nice rest of the day and a good weekend, and see you next week for our other modules every day next week. Okay, have a nice day. Bye-bye.